Welcome, and we're going to talk about our Comp Bio pre-college program today. We started our program in 2019 and had about 25 students in our first year, which we were very happy about. And since then, we've grown. So we currently have about 100 students a year, and that's going to be about the number that we have this coming summer. It's a great number because everybody comes in from across the country, across the world, and gets a chance to interact with each other and we sort of explore computational biology together. A huge part of what we work on in our program is group work and the opportunity for you to have an opportunity to work with each other as well as with teaching assistants as well as with us. And we love teaching our program. It's actually also expanded now to 4 weeks. So our program dates for 2026 are June 20th to July 18th. So we used to have a 3 week program. Now we have a 4 week program. And yeah, the sky's the limit. Maybe one day we'll just be teaching this year round. But uh, going to the next slide, as I said, my name is Philip Campo. I'm here with Josh Kangas. And Josh is going to talk a little about sort of his side of the program. I'm going to talk a little bit about my side of the program. The one thing that we always say to introduce our program is that we do a lot of things in our roles. Josh runs a master's program. I run our undergraduate program in computational biology. And we do a lot of teaching and administration within our department. But summertime is the best time because we get to teach this program. And it's always stressful and a ton of work, but it's always very much worth it because we get to just spend 4 weeks teaching some very bright students from around the world. And we also don't have to do the sorts of things that often bog down education, like giving midterms and grades and all those sorts of things. We just have a fun educational experience that allows you to learn something about a field that you probably may not know that much about. And we guarantee you that you'll know a lot more about what computational biology is and how it's basically the frontier of all of modern biology by the time you finish our program. So I'm going to talk a little bit about just general stuff and then hand it back to Josh. He'll hand it back to me. We're going to talk about a few different things here, but we're going to start with sort of, if you're interested in our program, what is it that we're looking for? So the number one thing is that we hope that if you're here or you're applying to our program, that you love science, and I'd say biology too. Although we've had students take our program, I think we've been able to grow the program enough that we've also started attracting students who are just smart and interested in science, and they may want to have a biology experience that goes a little bit deeper in terms of the biology itself, in terms of experimentation, but also in terms of analyzing data than what you usually get in a high school biology class. So if you're like me and you hated your high school biology class, computational biology is still might be the discipline for you. Part of why that is and why I love it so much is that it allows me to study science, but I'm also good at math and I really like to code. So if you have math experience, that translates to coding experience. We require one of those two things, but we don't expect that you've coded before. If you have AP Computer Science principles or A at your high school, then by all means, if you've taken those, make sure that we know about them. Or if you've done something else, let us know. But we still have quite a few students every year who are just strong at math, love science, and have never actually coded in their lives before. And that's okay too. So science and math are really what we're looking for. And then the third component here is not necessarily a technical component, but it just means that we want students who want to tackle something new because for each and every student that we bring to our program, we know that most of what we do is new to them. And having the mindset that you're going to spend 8 hours in a day working on something that you mostly have not seen before, we're looking for students who are like want to really lean in to those types of experiences. So we're actually one of multiple programs at CMU. We're one of the largest programs that are offered as part of the Central Pre-College Office. There are currently 12. I think next year there are going to be 14. And so we always give the plug to our other programs as well. On your application, we hope you'll apply to computational biology first choice, but you can apply to multiple programs and indicate what your hierarchy is in terms of which program you have first, second, or third choice. And we have all kinds of programs. They're not just in computer science. So we're one of the computer science programs, but there are programs in architecture, drama, design, writing and culture, and so forth. So check all of those out if you're interested in what we do at CMU, because I think there are a lot of really strong programs. And we have about 800 students across all of these programs at the university. About 100 of them are in computational biology. So we're one of the larger programs. One of the great things about this or any program is that you can stay in the dorms as a residential experience. And we know it's been cool when we check in with students to hear that, well, even though person X wound up going to a different university than I did, we still remained friends after that because we made such, strong friendships in the 4 weeks that we spent at Carnegie Mellon. So that's a critical component of our program, is that we're going to see you 5 days a week for about 8 hours a day. And then there's time outside of that that we really want you to have the opportunity to explore Pittsburgh and do a lot of the fun activities that the Central Pre-College office organizes, from baseball games to going to see the Falling Water Frank Lloyd Wright House to going to local museums and all this sort of stuff. But we also want you to just have fun and meet new people. So that's a critical part of our program and all of our pre-college programs as well. We have not yet published our tuition numbers for this coming year. Um, those will be coming out, I think, in the next couple weeks, or give or take. But we do want to stress, Josh, that we have scholarships available as part of our program. So we thus far, in 6 years of running the program, have been able to meet 100% of need in terms of students that have applied to our program. So there is a part of the application process that you can take part in that allows you to apply specifically to a scholarship as part of this. So we're a paid program, but we have scholarships available, and we've done well with that. We also have a few central goals of our program. So the number one thing that we care about is that you have fun, and both whether that's inside or outside of the classroom. Our classroom is set up to be fun too. And I always point to the number one, the top and the bottom point on this slide by saying, Josh and I strive to balance fun and challenge. It's easy to do one of those two things. You can go in and like just watch movies in a classroom, and that's fun. I've had classes like that in public high school, and you can go into a class and it can be really hard and very boring, but hard. So it's not hard for a college professor to make something that's hard. But what we're trying to do is balance those two things out. Where learning is really works is where it's challenging to the learner, but also where the learner is really enjoying themselves while they do it. And if we're going to bring you to campus and teach you for 8 hours a day, it better be fun, because I would hate to think that it wasn't. So we try to balance those two things out. I'll talk about what active learning here is shortly, but along the way, we want you to gain these skills that are like critical to what it means to be educated as a computational biologist. We want you to be a stronger programmer. We want you to appreciate that computation is everywhere in biology. A lot of times in biology, it's at this point in time, it's hard to find any aspect of biological research that isn't directly influenced or that doesn't immediately imply computational work. That doesn't mean that we don't think that experimentation is important. So the data have to come from somewhere, and we want you to get your hands dirty by engaging in real experimentation in order to generate that data that we're going to analyze. And that's a lot of what Josh is going to talk about. And we also want you to have an experience that shows the interplay between these two things. So you're going to do experimentation, you're going to become a stronger programmer. And then what the course is really about is combining these two things, and having the harmony between the experimentation and you writing literal code yourselves to analyze the biological data and get real answers to the questions that you have about real data. As I said, if you have not programmed before, that's okay. So you can take anything at your high school that's available, but regardless of where you come from or whether you've coded before, everybody has to do a coding boot camp. So on the next slide, I'll show you my banner here, and you can feel free to check us out. So this coding boot camp is hosted in part as part of my Programming for Lovers initiative, and it's actually an exciting time for it because we are launching a version of the course that's going to be in Python. So we're making the course multilingual, and our Python launch is going to happen on January 15th. If you would like to join us, you can go to our website and sign up for our mailing list. started putting a launch out a couple weeks ago, and we're at about 3,000 people who have signed up. So we're excited about that. Um, we'll also use a lot of these materials for the boot camp that you will do. So we'll have recordings of this, and we have auto graders that you can complete, and all this is self-paced. So you will know that you are expected to do this as of this moment in time, right? But if you're accepted to our program, we'll also send you information with details about exactly what you need to complete before you start our program, because we ask you to do a fair amount of work in advance, but that's all completely flexible. It can be done in your own time, independently, and we even provide like a teaching assistant who will guide you through it. If you have office hours, if you have questions, you can come to office hours and so forth. What's good about that course is it also is based off of scientific applications. So it's not just a coding course, and it's not just a Carnegie Mellon quality coding course that is very rigorous. It's also one that allows you to apply what you're doing to answer real scientific questions. We always start our program after you do the boot camp in advance. We start our program actually with some scientific exploration. So we've done a variety of different things in the Pittsburgh area. Here was our first year of the program, where we went out onto Pittsburgh's three rivers and we sampled river water from about 40 miles of rivers, which we then analyzed the bacterial composition of. That particular boat needs about $1 million, I think, in order to be fixed. So we've worked with a local nonprofit called Rivers of Steel and gone into some of the local ecosystems to look at, places where there are strange microbes. So if you happen to have $1 million on you, well, I'd say donate it to me. But if you have $100 million on you, maybe you have $1 million donation to Rivers of Steel, so we can get the boat operational. But we were doing a lot of other things. We have last year we went to an old iron furnace. We go to an environment in the Pittsburgh area that has acid mine runoff. So there are all kinds of wacky bacteria that are living in this very extreme environment. And that's been fun because Pittsburgh, in terms of like finding strange places where weird microbes might live, we are rife with that. After you do some environmental sampling, though, most days in your program are going to be divided into two halves. The first half of your day usually is devoted to a hackathon. And so this is where you'll work. I have a team of TAs, really that we always have absolutely exceptional TAs who are very dedicated and want to like see you succeed. And so each TA will be in charge of about 3 teams of 4 students. So you'll be in what we call hackathon pods of 4 or 5 students working together to solve questions that I pose to you. And I will do a little bit of lecturing in that class, but for the most part, I'd say most of your time, ideally all of your time, but sometimes I have to explain things, is spent working with your teammates in order to solve real problems and then take the code that you write and apply it to real data. So that's a fantastic setup because everybody wants to do well. And Josh and I last year even added like a Harry Potter style house contest. So teams that ask good questions or that perform well get points, and then there's awards at the end. The other half of your day, though, is with Josh, and it's devoted to the laboratory. So we can't write code to analyze biological data unless we get those biological data from somewhere. And he'll talk to you a little bit more about what laboratory techniques he applies and what you'll learn on the laboratory side of things here shortly. All of this is in keeping with our premise that the best classroom is an active learning classroom. And we point students to 2 articles that have been very well cited from the proceedings of the National Academy of Sciences, talking about active learning. And one of those says if you measure student performance in the active learning classroom, then students do markedly better. And you might wonder, what's an active learning classroom? Well, I think it's not going to be too much of a surprise given how our program operates. But the active learning classroom is one in which students are tasked with doing as much as possible instead of being tasked with being sponges for wisdom coming from the professor or the teacher. So we want you to get your hands dirty and run experiments. And some of you will get the experiments wrong. And that's good. That's a learning experience. We want you to come and code with me and work in hackathon pods and push yourselves to implement algorithms yourselves instead of seeing me do it. And what these 2 papers say is that classroom is way better for learning than a lecture room. And then the second paper actually says a lot of times when you poll students and say, do you feel like you're learning? A lot of times students will say, yeah, I wish I was in a lecture. I feel like I would be learning more. And that's because those students are wrong. Um, a lot of times, if you're in an active learning classroom, you're going to be pushed and challenged, and it's going to feel hard, and you're going to think, man, this is tough. And we always tell students, if you ever have an experience like that, that's good because that is what true learning feels like. I'm going to hand it off to Josh. He's going to talk about what our big topics are in terms of like what we look at from a data perspective, why we're capturing data, and then how it is that we're going to extract those data experimentally.
All right, thank you. So, Josh Kangas, teaching faculty in the Comp Bio department. What really excites me about this program is the opportunity to teach students about the interface between the computation and the analysis that we can do and how we generate the data. And what's super exciting is the choices that are made in the context of the data generation in the lab have major impacts on the kinds of computation you can do and the algorithms you might apply for the analysis. So all of the courses that I teach involve that fundamental idea where we're looking at the interface between lab and computation. So we have some big topics that we cover. One of them is that we look at the Pittsburgh water and soil microbiome. So we go to a few different locations in Pittsburgh and collect samples and try to understand, what are the bacteria that are present in these environments. And we attempt to learn relationships between these bacterial populations and specific aspects of the environment that they live in. So as an example, we have these two rivers that we're showing here. On the left side, it's the Allegheny River. On the right side is the Monongahela. And they come together at the point, which is at the bottom in the center here. And then they form the Ohio River. So these three rivers that are differently named, but from two different sources of water, they have very different environments. And so we want to study those environments. And then when we're looking at Boyce Park, we're looking at the acid mine drainage ponds and trying to understand what bacteria are thriving in those environments. And those environments are related to the history of the area where there's been lots of mining, which causes the release of very acidic water when these mines fill up with water. And then as Philip mentioned, we also went to Cary Furnace, and we're looking at the microbiome in soil in the area of these furnaces that were used in the production of steel. So lots of interesting topics that are locally relevant, historically relevant, and computationally interesting. Yeah, the next big topic is this pandemic, right? I think you've heard about this. So we can actually study the SARS-CoV-2 virus. There was during the pandemic and now ongoing, there was a lot of sequencing efforts to understand the source of the virus, as well as how it mutated over time, and that gives us a great opportunity to study an event that had a huge impact on all of our lives, but also for which there are really interesting biology questions that require computation to answer them. And so we'll spend a lot of time focusing on them. We will not do anything, hopefully, with the virus in the lab. We've mostly avoided that so far. Okay, so in order to generate this data, we need to cover some topics in the lab. So we want to give you kind of a brief summary of the sorts of topics that we're addressing. Sorry. So first, we do environmental bacterial sampling. And so we will take samples from water and we'll take samples from soil, and there are different strategies for doing this. We'll do transfection. So transfection is the process of starting with a piece of DNA called a plasmid that codes for a gene and inserting that gene into bacteria to cause a change in the bacterial phenotype. We'll talk about how do you isolate bacteria. So if you have a superbacteria, how do you get one single species of bacteria all by itself? Here at CMU, we are very interested in automating science. And as part of that, we're going to show you how to use liquid handling robotics where we have robots that will actually do pipetting for you. So they'll be moving liquids around instead of having to do that by hand. We'll also do some bioart, and that will be both robotic and manually, if you would like. We'll do DNA extraction. So if we have this soup of bacteria and we want to identify what's in this soup of bacteria, we need to get the DNA out. And because the bacteria have kind of complex structures and different structures across different species of bacteria, we need a robust DNA extraction strategy so we can get that DNA out and purify it. So then we can do more experiments on that DNA. One of those types of experiments is called the polymerase chain reaction, where we're making lots of copies of DNA in a very careful way. We'll also do sequencing. And so we have different sequencing methods that we're going to use in the course. So Sanger sequencing, Illumina sequencing, and then Oxford nanopore sequencing. And we'll talk about the differences between those and when and why you might want to apply one method over another. We'll also look at the spatial growth of bacteria, so we can visualize how bacteria are growing in a horizontal surface. We're going to do some artificial selection of extremophiles. We're going to try to find very rare bacteria. One of the challenges for microbiome studies is that we can't always isolate bacteria, but we can get the DNA and at least identify that it's present. So it's an additional challenge to isolate individual bacteria that thrive in these really weird environments. So we're going to work on doing that. We're going to do gel electrophoresis. We're going to characterize bacterial colonies. If you've ever done any kind of bacterial isolation from an environment, you get plates that look like what we have here on the bottom right. And the bacteria are going to show up in colonies that are different sizes, different shapes, different colors, different textures. And so we're going to characterize those colonies. We're also going to quantify DNA just to figure out how much we get out of our extraction processes, and also try to determine the quality of the DNA that we extract. So these are all key lab techniques that go into molecular biology and microbiology experiments that we're going to teach you and you're going to experience in the lab. I also mentioned that we have this interest in automated science, and I wanted to take a moment to kind of describe what that means and how you might experience that or how you will experience that in this course. So basic biological research, if you want to think about what goes into that, you have a lot of manual pipetting, paper protocols that are pretty fixed and rigid, where you're doing all this work. We'll do some of that because it's necessary as part of building your experience. But then if you want to take that kind of biological research to the next level, you can do high throughput biological research, which is where you're trying to generate as much data as possible, as quickly as possible. And you can use robots to help you generate those data. So you can miniaturize experiments and run lots of them concurrently to generate mountains of data. But then the next phase, so automated science, is where you're using AI to choose what experiments you're going to run, and then the AI can tell a robot what experiment to run. So using AI to drive the experimentation process. And you might say, oh, that's crazy. But it's not actually that crazy. So you can frame certain problems in a way that you can use algorithms to begin to address those problems in a very efficient way in terms of the data generation. So as an example, when you're playing the game Battleship, if you're familiar with that, you take individual shots on a game board. And every time you take a shot, you learn something new about your opponent's game board. And then you make decisions based on what you have learned. And so you can convert that into an algorithm. So if you can frame a biology problem in a way that's like the game Battleship, then you can write an algorithm to solve it. So we're going to talk a lot about that. We're also going to have a Battleship tournament because we can use robots to play Battleship in a chemically oriented way. And so we'll have a tournament, and you'll get a chance to compete with each other to see who can come up with the best AI. And we'll probably also have some guest AIs that are included in the mix as well. So we're going to use automation, machine learning, and AI to learn models about different growth conditions as well. So we'll be learning the complexity of uh, what bacteria respond to what environmental constraints. And that'll be very interesting and a nice introduction to basic machine learning. You're also going to get a chance to see my lab. So my lab is in the top left here. And in that lab, we have a robotic arm that will move plates around between different devices. And those different devices, manipulate experimental samples and or take measurements of them. And so we'll show you that in process. And CMU is also the home to a cloud lab. And the bottom right-hand corner, we have a picture of our cloud lab. And so we will do our best to get you an experience to see inside of that cloud lab and perhaps use it if possible. Okay, so what do we do after we sample the water? That's an important question. So the first thing we do with the water samples is we filter out the biological material. And that means like bacteria, viruses, plant matter, all kinds of stuff. And then we extract the DNA. So we break open all these cells and pull the DNA out, and then we analyze the DNA. And so the kinds of analysis that we're doing would be sequencing. So with sequencing, we can break the DNA up into little tiny pieces, and then we get A, C's, T's, and G's representing the sequence of these pieces of DNA. So then the big question that we're going to answer computationally is what can we do with this kind of sequencing data, and what interesting algorithms can we apply to help us understand and answer important science questions from sequence data. So with that, I'll turn it back over to Philip.
Thank you, Josh. I'll try to keep things reasonably brief in terms of what are the big picture questions that we want to answer computationally. So Josh has talked a lot about bacteria everywhere, even in your body. Over half the cells in your body are bacteria. And there are a lot of human bacterial studies that we might want to do. So, like, for example, just studying the bacteria that live in your gut is still a problem that people are working very heavily on and that we really are only starting to unlock understanding of. Like, a really interesting fact I only learned relatively recently is that something like 90% of the serotonin that's made in your body is actually made by bacteria in your gut. Which you think of that as a brain chemical, and it is really important for your brain health. But it actually, so much of that is coming from cells that are not even yours. And so when Josh talks about like what we're getting from this is this DNA stew, a huge first question is to say, well, how is it that if I take one sample and compare it to another, maybe it's the same place in the river over multiple years, or it's two different ponds that we sample at the same time. How is it that we're able to actually differentiate these? Like, how diverse is each sample? How different are those two samples? And all that relies on us taking these DNA stews and comparing them. And it means that we have to come up with like quantitative approaches for doing this. And so that's what we call metagenomics. Like we've got this DNA or RNA that we sample from the environment, and then we want to figure out how is it that like bacteria in that environment change over time or sample to sample, and all that just depends on math and programming. The second thing that we might do is to say, well, once I know what those species are, how can I compare them? So in module 2, we look at things like sequence alignment, algorithms that allow us to compare, for example, two genes, the same gene from two organisms, or two entire genomes, which we do for viral studies, or comparing, say, one small region that we've captured from a sample and say what genome of what bacterium does this actually come from. So all those questions ultimately rely on making comparisons of strings. And if you take our problem, you will see this figure on the right until you get sick, because it turns out we can frame this idea of string comparison as a problem that is equivalent to finding the longest path in a network. And so we wind up doing a lot of algorithms around network analysis in order to figure out how to answer this question that's a biological question. In module 3, we look at something a little bit different, and this was inspired by us looking at both wanting to come up with full genomes from the samples that we were taking, as well as something like SARS-CoV-2. So there's a critical question that's central to modern biology, which is how is it that you figure out what the genome of a species is. In our case, it's 3 billion letters. For a bacterium, it might be like 5 million letters. For SARS-CoV-2, it's 30,000. How do they know what that sequence of letters is? So this is the culmination of literally decades and billions of dollars of research into two aspects. One, being able to take multiple copies of a genome, which in your case, for example, might be getting a blood sample and then isolating the white blood cells, which all have the same DNA, fragmenting that into smaller pieces called reads, feeding that into a sequencing machine that then feeds out these are the identities of the nucleotides in each of these fragments. And then using the fact that we started with multiple identical copies, assemble the genome, which is shown here as the black string at the bottom, by seeing that, well, the orange fragment overlaps with the blue, which overlaps with the teal one, and we're able to use that to infer what that genome is. It's like a puzzle, except the puzzle has hundreds of millions of pieces in the case of humans. And even in the case of a virus, it will have tens of thousands of pieces. And we can't simply do this by hand. So we need algorithms in order to do it. And we learn about what algorithms those are. In module 4, once we have a genome, then what we're trying to do is basically combine modules 2 and 3 here to say, once I'm able to make comparisons of things using algorithms, comparisons of genes, so to speak, and once I know what the genomes are, how is it that I can what's called annotate the genome, which is determine where it is that the genes within that are lurking, and so that I can know what they do. This often is a database search problem too, because once we start answering this question, we can use similarity to say, well, this is probably what this gene does in SARS-CoV-2 because we've studied coronaviruses for many years, and these are similar genes that we already know the function of. We'll see how machine learning is useful in that context as well. And then in the final module of the class, we look at evolutionary tree construction. And this is really cool because what we do in the class is a couple things. One, we can take SARS-CoV-2 viral data sets from around the world, and we can show you here is how the virus has mutated as it spread through the human population, and how is it that they knew what viral variants are. So when the next epidemic hits, we'll have once again real-time genome sequencing, and we know how to find new variants as they arise. This is a central problem that goes back to Darwin's journal in 1837 of forming an evolutionary tree. And the question is, how do we use that DNA data in order to feed that into some algorithm that's going to spit out what we hope is a correct evolutionary tree for this collection of organisms? So yet another computational aspect of biology. And then we'll also do this for the bacteria that you sample. So we always have like a fun aspect of the course where everybody has like one bacterial species that's like theirs that they identified, and we'll sample a fragment of one of its genes and sequence it, and then we build an evolutionary tree of that. So at the end of the program, you'll know this was the identity of my sequence because we were able to apply our algorithm to know what it is. And then we'll build a big evolutionary tree so you can see here's the evolutionary tree of our pre-college program with what each bacterium actually corresponds to. That's what we do on the hackathon side. And at each point, as I said, you're going to be solving like dozens of code challenges in groups in order to solve these questions. I'll hand it back to Josh. Josh is going to wrap up. He'll tell you a little bit about some student testimonials, handle some FAQs, and then say a little bit about our application. And then we're happy to answer your questions.
All right. So we have a few different student testimonials. One of the important parts of this course that we do every year is that we build these pods of students, and we're very careful in the way we build it. We have an algorithm that constructs very diverse groups of students. So you might be working with someone from a very different part of the country, yeah, and maybe they have different languages, different high schools, whatever. And so we emphasize the diversity in the group building. And this student is specifically talking about how she noticed this. We didn't advertise it the first year we did this, but she noticed it right away and noticed the value that that brought to the discussions that she had in her learning process. We have student Erica. She is she was in the first cohort of the pre-college program, and she's basically making the point that this helped to prepare her for PhD studies and kind of set her direction. She was the first pre-college student who got into a PhD program, and she just started, I believe, last fall, right? And so we're excited for that, and this kind of gives you a sense of where our students go longer term. Rayash is one of our favorite students. So he was also in the first cohort, and he was a great student. And then he went on to become an undergrad at CMU. And then when he finished his undergrad, he stayed on for a master's degree. So he just finished last in May, and we miss him a lot, but is one of our success stories from the pre-college program, one of many. Uh, Eliza was from the class of 2024, and she is basically talking here about how she hadn't coded before. And this course set her up with a great background in programming. And she's now a very strong programmer. And she just is emphasizing that, you know, any student can thrive in this program because we take students with different backgrounds, and we really work hard to make sure that the experience is great for all of our students. Based on the efforts from Philip and me, and then our small army of TAs that are essential to this process. So we have a few FAQs we wanted to go through. So the first one, do we require standardized tests? So we don't require standardized tests, but they can be helpful for your application. Philip mentioned the characteristics that we're looking for. So standardized tests can help us see your capabilities in math. So you might include them, but they're not required. As far as the recommendation letters, we're looking for recommendation letters from teachers, research advisors, if you have them, and counselors. They're all good. Science teachers, math teachers, computer science teachers, those are all great. If you live in Pittsburgh, you're welcome to join us, and you don't have to stay in the dorms. We usually have a few commuters, one or two commuters a year, that don't live in the dorms, but they get to experience all of the same social activities as our residential students. They just don't physically stay in the dorms in the night. And then, how old do I have to be to join the program? So we have a very rigid requirement for legal reasons that we don't establish, but you have to be 16 on the first day of the program in order to join us, and there are no exceptions to that. All right. So to wrap up, we hope to see you in 2026. The URL for the application is here. The tuition is going to be posted soon. The application will be open soon. The early deadline for scholarship international students, you need to get your application in by February 1st, and the regular deadline is March 1st. And so we hope to see your applications, and we're really excited that you joined us for this information session. I hope it was useful.